So kind of strap in. This is going to be a bit of a long one. I'm also going to put timestamps at the side. So if you're missing maybe just like a part of this, you can skip ahead to that timestamp and you don't have to see stuff that you've seen already. Also, word to the wise, you can watch lectures. You can watch any YouTube video actually sped up. So I would recommend just clicking watch at 2x speed maybe and get to the part that you aren't quite understanding. But here we go. In this lecture, we're going to be reviewing the structure of DNA, which is going to become important at the very end. We're also going to be reviewing restriction enzymes, which also is going to become important at the end. And we're going to talk about PCR again, and we're going to talk about the enzymes that are involved in replication, which will be a good review for the final exam if maybe you aren't quite up to speed. I've also made separate videos on each of these topics, so again, this is just stitching everything together. Feel free to go back to those previous videos. But let's talk about the structure of DNA. Here's a complicated looking picture. Why don't we simplify this down just a smidge? And let's zoom in on this part of it. This is still fairly complicated, so let's get a little bit simpler. There we go. What I want you to know about DNA is that it is made up of this backbone of repeating phosphate and sugar subunits. So here's our phosphate, here's our sugar. Here's our phosphate, here's our sugar. Okay, you don't necessarily need to know the chemical composition of these things. You should know that there's a bunch of carbon in here. And you should also know, again, that it's a phosphate sugar backbone. The third thing to sort of know is that these sugars are connected to bases. They're connected to like base pairs, right? Those are your A's, G's, C's, and T's. And so there we go. There we have our anti-parallel complementary strand on the right. And you'll notice that our sugars and phosphate groups are kind of like flipped upside down because, again, it is anti-parallel. They're heading in opposite directions. So you may have heard me say, and here's this all put back together, you may have heard me say in the past that DNA is negatively charged. The reason that DNA is negatively charged is because of these phosphate groups. So remember from 5 seconds ago, it's a phosphate sugar backbone that makes up this structure of DNA. But these phosphate groups are negatively charged. Okay? These phosphate groups are negatively charged. And that gives DNA a lot of really interesting properties. But we're going to exploit just one of those later on. I don't necessarily need you to know those properties, but I would like you to know DNA is negatively charged, and it's probably a good idea to know that it's negatively charged because it has a bunch of negatively charged phosphate groups. Okay, so we've done a brief review of the structure of DNA. Let's talk about restriction enzymes for just a minute. What are restriction enzymes? What do they do? Restriction enzymes cut DNA at specific places, which we call restriction sites. So restriction enzymes, their entire job is to cut DNA at these restriction sites. Remember, we found these interesting little enzymes in bacteria, and we figured out that what they do in the original bacteria is they help them fight viruses, because there are a lot of viruses that kill bacteria. And so bacteria evolved these restriction enzymes to help them fight those off. And we stole them so that we can use them for some of the cool biotechnology, one example of which I'm going to show you in just a second here. So again, restriction enzymes cut DNA at specific spots called restriction sites. So here's a restriction site here, which is maybe a little bit overly complicated. So let's simplify things a bit. Is that any better for you? Probably not, but you can still see the phosphate sugar repeating backbone. Let's get this even a little bit simpler. Again, you have the phosphate sugar backbone with these bases here, but this is probably too much for us still. There we go, that's a little bit better. So this is a restriction site. Okay, we could even simplify this down again if we wanted to look like this. So the restriction site EcoR1 is GTTAAC. Anytime you have that segment of DNA and you have it with a restriction enzyme, the piece of DNA is going to get cut. All right, that is all the restriction enzyme is supposed to do, is cut DNA. So anytime you have this particular segment, or if we had it, again, double-stranded, it's going to get cut right there. We know that. We've studied it. Interestingly, this won't be on the test, but interestingly, you should notice that this is a palindromic. Segment. So it reads the same forwards as it does backwards, like race car. Anyway, it's GAATTC forwards and it's GAATTC backwards. So restriction sites are most often these palindromic sites. Just interesting, neither here nor there. The point being, again, anytime you have this segment and you have the restriction enzyme EcoR1, your DNA is going to get snipped. It's going to get cut right there. Which is really cute. That's fantastic, Aaron, but who cares? Why do I care? Why should you care? Why does anybody care? We care because let's say we have some gene that looks like this. And what I'm showing here is that it's a gene that is flanked by restriction sites. You have restriction sites on either end of this gene. Because we have that, we would be able to cut this gene open or cut this gene out and remove it from a segment of DNA. Okay, because we're able to cut these genes out, that allows us to analyze the DNA, it allows us to modify DNA, and it also allows us to transform DNA in other organisms. So again, what does this look like to you? Hopefully this looks a little bit familiar. What is this circular piece of DNA here? This is a plasmid, and because we are really good at manipulating plasmids, let's say you had a restriction site here, right after the promoter, and a restriction site here. That would allow us to cut that gene out and use it for other things. It would also potentially allow us to paste another gene into the plasmid where we want. Okay, so restriction enzymes are a really big deal in biotechnology. They allow us to do a lot of really cool stuff. It also allows us to analyze DNA. We can analyze DNA without restriction sites, but it certainly helps with this process. And we're going to talk about how. So the next topic here is PCR. And what are we doing when we PCR something? Like, what are we trying to actually do with that? Well, what we're trying to do is we're trying to make a copy of DNA. We're trying to basically steal the molecular mechanics for replication of DNA and use it for our own means. So again, when we do PCR, we are replicating DNA using our own cleverness and ability to steal. And there are three main enzymes. There are more enzymes in this, but there are three main enzymes that we discussed when replicating DNA. There's the helicase enzyme, there is the primase enzyme, and there is the polymerase enzyme. And so helicase melts the DNA or causes the hydrogen bonds between those base pairs to become unbonded and opens it up and allows polymerase to basically make copies and extend that segment of DNA. But what we have done is we just use heat. We use heat to open up that DNA. And because we're using heat, it allows us to open up the entire segment of DNA. And then the next enzyme that's involved is primase, because remember, polymerase doesn't know where to start. If this is all foreign to you, go check out that previous lecture from before. But helicase, we're using heat as our helicase, basically. And then primase would normally come in and add primers so that polymerase would know where to go. But because we're adding in our own primers, we don't need primase in this case. We can just replicate the segment of DNA that we care about. The final enzyme involved in DNA replication is DNA polymerase. And again, because we're heating up our DNA to get so hot, we have to use that heat stable polymerase that we call Taq polymerase. Taq polymerase is polymerase from the bacteria Thermophilus aquaticus, the extremophile. That was a question in the lab. Why doesn't our polymerase get disrupted or get denatured by the heat? And it's because we use heat stable Taq polymerase. Pretty cool stuff. So here's a picture of PCR in action. Here is our template copy. We denature it, or we heat it up. We toss in our primers, and then our polymerase makes a copy. Second stage, we melt it, throw in our primers. Well, our primers were already there. Polymerase extends, and we have more copies. And you just continue this cycle of this polymerase chain reaction, and at the end you wind up with many copies of, even if you just start out with one segment of DNA, you still wind up with 10 to the 9th, which is like, I don't know, a billion or something like that, after just 30 rounds. So we talked about, again, the mechanics of that in a previous lecture, but the big picture is that PCR is going to leave us with many copies of a segment of DNA. Whatever segment of DNA we care about amplifying, that's what we're going to be left with. Which is great, that's cool, but who cares? Why do we want a bunch of copies of DNA anyway? It's kind of gross, right? Like, why do you need a bunch of DNA in a tube? What difference does that make? The reason that you want to be able to have all of those copies, or the reason why it's valuable to have those copies, is because we actually figured out that we can analyze them and figure out what DNA we're actually looking at using my friend and yours, gel electrophoresis. So, quick overview of everything that we've talked about. DNA, negatively charged. Why is it negatively charged? It has those phosphate groups. Restriction enzymes, they cut DNA at specific locations. Okay, and we know what those locations are, and there's different restriction enzymes that cut at different places. PCR, it makes many copies of a particular segment of DNA that we care about. Basically, anything that's past the primer. So you want a segment of DNA copied, throw in a particular primer, and you're going to get everything downstream of that. It's called downstream. But you're going to get everything downstream of that primer copy. Okay? What are the enzymes involved in DNA replication? Helicase. Well, how do we mimic helicase? We heat up the DNA. Primase is involved. How do we mimic primase? We toss in our own primers. Polymerase is involved. How do we do polymerase? Well, we just use regular old polymerase, but it's polymerase from a thermal stable bacterium, Thermophilus aquaticus. Okay. Now that we've got all that under our belt, let's move on to a super cool thing called gel electrophoresis. So what is gel electrophoresis? It's a technique that allows us to separate DNA based on the size of that DNA. And I threw this picture in. This is actually a pretty poor gel because there's all these streaks going down. This looks like a gel I would have done. But let's talk about how that actually works. So this is what an actual electrophoresis machine looks like. And these things are, they're not crazy big. You can get them to be pretty big if you want to run some like crazy big gels. But they're generally pretty small. You could fit one in your backpack super easily. Here is the gel itself. Here are your two electrodes, your negative electrode and your positive electrode. Here are your wells. This is where you're actually going to pipette your DNA into. Here's buffer solution. You don't need to worry about that. But so, what's the charge on DNA? DNA has a negative charge naturally, right? Because of those phosphate groups. So if we apply electricity to some DNA, where is it going to go? Well, opposites attract, likes repel, and so just naturally the DNA is going to move from the negative end toward the positive end. And so the thing about this gel, it's called agarose gel, and basically it's kind of like an an obstacle course for the DNA. You could think of it like that. And so based on the size of the DNA, it's going to travel through this obstacle course at different rates. And so the smaller, quicker, faster DNA is going to be able to move more fast through this gel. And so your smaller bands of DNA are going to be down here. Your larger bands are going to be up here. So this is kind of like an application problem. By the way, this is not necessarily how this analysis would be done. I just wanted to tie a whole bunch of loose ends together and sort of put some different things together so that we can figure out how you would actually analyze a segment of DNA. So let's say you're asked to determine the paternity of a child so that we can figure out who's going to get an estate or something like that. Actual reason isn't important. You're given cells from 3 prospective fathers and from the kid, the child in question. You only have a little bit of cells to work with. Maybe the warrant wasn't well written. Maybe the fathers didn't care to participate. And we know of a gene that has a restriction site that we can use for analysis. So what's the first thing that we should do? Well, we have cells, so we should probably extract the DNA from them. So we need to get the DNA out of the nucleus and out of the cell membrane. Well, how do we go about getting DNA from inside of a cell into inside of a tube? You use detergent and mechanical methods, shake the DNA. You can also do things like sonicating, which is basically just putting sound waves into it. And you can, apart from just gently shaking, you can put them into a machine that will shake them, and you can let that run overnight. We're going to use salt also to prevent that DNA from reacting with the water. That's probably the step I'm least concerned with. And we're also going to use alcohol to precipitate our DNA. Okay, DNA will not dissolve in alcohol. So maybe you remember from way back once upon a time when we were talking about chemistry that like dissolves like. So water will dissolve DNA. Why will water dissolve DNA? Well, water is polar, hopefully you remember, and as we've been just talking about extensively, DNA is polar as well. Whereas alcohol is nonpolar. DNA is polar, so alcohol will not dissolve DNA. If you take a bunch of DNA and you put it into alcohol, the DNA is going to do what's called precipitating out. And as you guys saw, it's going to look like a little white piece of mucus on top of your solution. So we use alcohol to precipitate the DNA because DNA does not dissolve in alcohol. Cool. So we've got some DNA. That's fantastic. Well, now what are we supposed to do with it? Let's go ahead and freeze most of our DNA, which we have in these larger tubes here for dad (1), dad (2), dad (3), son, and our negative control. This negative control, by the way, would just consist of basically whatever buffers and whatever solutions we use for all these other steps, but should not contain any DNA. But anyway, I'm getting off topic. Let's go ahead and freeze most of this. So we're just going to toss these containers of our DNA samples into a freezer. This is what a -80 freezer looks like. You could store DNA in just a regular freezer, but it's more stable the colder you get. And also, if you just open the freezer up, your regular freezer is going to get really, really, really warm and possibly disrupt your DNA. So we're going to toss our DNA, our original DNA, into this -80. And you might be asking yourself, well, why do we freeze our samples if we already have the DNA? Like, why do we need to freeze it? There's a few different reasons. The main one is because scientists make mistakes. Very human, even very simple mistakes get made kind of all the time. The other reason is if we find our test to be inconclusive somehow, we're going to want to be able to go back to that original piece of DNA and resample it, and hopefully be able to run more tests on it. Also, if we had enough cells, those cells would be frozen in the Midas 80 as well. So we've got our DNA here. We've got it sort of subsampled. But now how do we know that we have enough DNA? How do we know that if it's high enough quality? How do we know that we have enough that we're actually going to be able to run PCR and get the copy of the DNA that we want? And so this probably isn't going to be on the test. We need to figure out how good our sample is. And so you use a little DNA analyzer here. This is called a qubit. And it's going to tell you, for one thing, how much DNA you have, and it's going to tell you what is the quality of DNA that you have. And so it's going to say, well, you have, this much DNA per milliliter, microliter probably, and it is of reasonably good quality. This is kind of a new technique. They used to just use fluorescence, and basically what they would do is they would shine a light into your DNA sample, and they would say, well, DNA absorbs light at this spectrum, and so you have enough DNA probably. These new advanced machines actually interact with the DNA chemically and emit light, and they are very cool, very fantastic, not crazy expensive, surprisingly. But let's just assume then we have high enough quality DNA. So the next thing that we need to do is we want to PCR amplify the particular segment of DNA that we care about. So what we're going to do is we are going to take our tubes and we are going to throw them into this thing called a thermal cycler. So you probably remember from before, the process of polymerase chain reaction involves heating up your DNA and then letting it cool back down, and then heating it up again so that you can melt those segments of DNA, and letting it cool back down so the polymerase can extend, and then heating up your segments of DNA. That's what a thermal cycler does. Okay, it cycles through those processes of, not processes, but it cycles through those levels of heat so that your DNA can be copied. And these take about 90 minutes. They take about an hour and a half. Most people that I know just start them before lunch, or they start them at the end of the day. And then when you come back to the lab, you have a setting on there that will cause your DNA to actually deep freeze, so that it stays true and all your copies are preserved. Your DNA won't begin to degrade by just sitting around at room temperature. But they take about 90 minutes to run to give you billions of copies of DNA. Great. We have some copied DNA, pull these things out next morning or after lunch or whatever. So what is actually in each tube? So what should still be in each tube is probably several segments of your original genome. Those are still going to be in here. But those are going to be absolutely overwhelmed by the millions of copies of just our segment of DNA that we're interested in. So whatever was immediately downstream from our primer, there's going to be millions and millions of copies in each and every one of these tubes. So we know that if an individual has a variant of the gene that we care about, the segment of DNA that we amplified will be cut into 4 bands by ECO R1. If the person does not have that variant, it will be cut into only 2 bands. So now, again, what's in each tube? Well, it should just be the segment of DNA that we amplified. And it should all be the same whether or not the individual has that gene. So let's take a look and just think for a moment. So like we just said, if they have the gene, it's going to get cut into 4. If they don't have the gene, it's going to get cut into 2. Let's talk about that for a second. Why does this happen, that these things are cut differently? Well, it's all just segments of DNA. So here's like what the person with the gene, this is what their DNA looked like. The person with no gene, this is what their DNA looks like. You can't really differentiate them until you realize that there are these restriction sites. So the person with this gene has these restriction sites. The person without these gene has this one restriction site. And if you were to subject this, or if you were to look at it very, very closely, if you were able to sequence it, like let's take a look at this spot in particular, what you're going to find is this exact segment of DNA, GAATTC. In fact, if you look at all of these, you're going to find that exact segment. You're going to find that exact restriction site, GAATTC. Why do we know this? We know this because we've sequenced this already. Why do we care about this? We care about this because it allows us to tell who is who, or who is related to who. So let's go ahead and digest everything with EcoR1. How do we digest things with EcoR1? You pretty much just pipette it in. You add your EcoR1. There's a few other steps, but that's mainly it. It kind of just does its thing, mostly on its own. It's going to cut up that DNA. So now what's in each tube? What is in each of our tubes here? Well, that kind of depends. If the person has the gene, you have 4 segments of DNA. Actually, you have millions of segments of DNA, but when you run it on gel, you're going to find 4 bands. If the person doesn't have the gene, you're only going to find 2 bands. So let's talk about what the rest of this process is like. Let's run our DNA on a gel. So if your lab is rich, you have these gels purchased and brought in for you. If your lab is poor, or if your PI is a bit of a jerk or whatever, they make you pour your own gels because it's much cheaper to do it that way. Again, these gels have these wells at the top where we can add our DNA. So let's go ahead. We're going to add our DNA that's been PCR amplified and restriction digested into this lane here. We're going to add DAD2 into this lane here. DAD3 goes here. Control DNA. Oops, I missed sun DNA, didn't I? Control DNA goes here. Our ladder DNA goes here and here. And we'll pretend like we went ahead and added the sun's DNA, even though I didn't show that. Let's go ahead and apply our electricity using our PowerBox 3000 thing. And let it run for a bit. Or if you're like my friends, you jack up the voltage all the way and you let it run for about 5 minutes, and it's ugly, but it usually works. Anyway, so we've got our gel here. This is what it looks like after we've run everything. And we ran 2 ladders, by the way, just mostly because we had an extra lane over here, and it can make things a little bit easier to visualize. Sometimes people will put one here and then they'll put one on the middle. Sometimes people will just put one ladder. It doesn't really matter. We just ran it this way because we had an extra one. So which segments of DNA are smaller? Where are the smallest pieces of DNA? The smallest pieces of DNA are down here on the very ends. So those are the tiny pieces of DNA. And the largest pieces of DNA are up here. So let's start by asking who has the gene that we're interested in. Well, DAD2 has 4 bands, 1, 2, 3, 4, and the sun has 4 bands, 1, 2, 3, 4. So DAD2 question mark and the sun each have 4 bands. DAD1 and 3, you guys can go home. You are not the father, conclusively. But using this technique, can we say that DAD2 is the father for sure? Well, no, not really. We just know that DAD1 and DAD3 are not the father. Further analysis would be needed for that. Again, we can eliminate DAD1 and DAD3. So this is a technique called terminal restriction fragment lake polymorphism. It's not used very often. It was just a technique that I thought was pretty good that we could tie everything together with. And you could see probably from all the many tedious steps and the lack of conclusiveness why it might not be used. But it is used occasionally. and it was used more frequently before. Let's talk about what we just talked about. We talked about the structure of DNA for like the 100th time. It is a repeating sugar phosphate backbone. The phosphates are negatively charged, which allows us to do electrophoresis. We talked about restriction enzymes, which cut DNA at very specific places, which allows us to not only analyze those segments of DNA, but potentially paste in segments of DNA where we want them. We talked about the polymerase chain reaction, which is just a molecular copy machine. And we talked about how we've been able to mimic or steal those replication enzymes that DNA uses to replicate in cells. And we also talked about gel electrophoresis and how we can use it to analyze DNA for forensics. Okay, that was a marathon. Again, always let me know if you guys have any questions. Keep those emails coming, and I will see you guys online.